The topic for today is immunology, and the speaker that we have here is Dr. Saibal Moitra, who is a professor and senior consultant at and allergy and immunology depth of pulmonology in Apollo Multispeciality Hospital, Kolkata. Sir, you may start now.
Yeah, thank you. Okay, so good evening to all. So this is actually a brush up of the immunology, and I would just go through the basic tenets of the immunology, which you need to understand and know while you are dealing with any means disease or anything of that sort. Because once you should have a fair knowledge of the immunology, which actually helps you to understand the pathomechanisms behind the various diseases, whether it's autoimmune condition or an allergic condition, or for that matter, cancer, or means any inflammatory conditions. Okay, so means after the class we can have a Q&A session, and maybe if you want we can have another revision class of immunology. So the objective of this lecture is to provide fundamental concepts on the immune system. It's for the general medical audiences who are aspiring for the MRCP exam. Okay, so you see there's many things inside. No, this seems to be a very busy slide. Okay, with lots of things scribbled around. So seems to be a complex slide. This is what our immune system is. Immunology is a complex subject. So it's a network, okay? And in the web of this is system, there are many things which goes on occurring, okay? But there is always is a tandem inside it, how it functions, how it works. And there are some basic principles on which our immune system works. We'll try to understand the basic principles, and then you will see that all these busy diagrams which is there, it would not appear that busy, and you would be able to correlate it in any disease conditions. So I would discuss mainly the origin of the vertebrate immune cells, that is number one, then the principles of the innate immunity, the principles of the adaptive immunity, and the effector mechanisms of the immunity. So our immune system is a defense system. Right, we have this panoply of means millions of organisms outside us. It could be a virus, bacteria, fungi, protozoa, or maybe multicellular elements. So which is there to attack our body. So our body needs to defend itself from all these organisms which is around. So you see, it's a hostile world, and to live in a hostile world, we should have a strong defense mechanism which is going to prevent us from getting a disease or getting degraded. So why do we have an immune system? First of all, we know it's a defense system, so we need to recognize the pathogens, which is non-self, and to organize a defense response to any pathogens which enters our body. And at the same time, it helps to destroy and eliminate the pathogen which has entered the body. So this is one of the functions of the immune system. So other functions of the immune system we all know is the immune surveillance. Immune surveillance is it helps to detect any rogue cells which have come up inside the body. So it detects as that rogue cell immediately and kills it off and prevents it from multiplying and causing a disease in the host. So that means immune surveillance, which is very, very important in diseases like in cancer, where there is the suboptimal immune surveillance leads to the growth of cancer cells. And there are various ways in which the cancer cells can bypass this immune surveillance mechanism in an individual. Okay. So these are the main purposes. Now, where do these cells originate from? So when we talk about the immune system, it's not only the cells, that is the lymphocytes we are talking about. So all the cells which comprise, which takes part in an immune defense mechanism, they all come from the hematopoietic stem cells in the bone marrow. So that is The bone marrow or the hematopoietic stem cells give rise to all the cells which takes part in the immune defense mechanism. Now, so what happens is that we know very early in the maturation of the cells, the metabolic stem cell, it divides into a common lymphoid progenitor and a common myeloid progenitor. The common myeloid progenitor then gives rise to a granulocyte or the macrophage progenitor, and it also gives rise to a megakaryocyte or erythrocyte progenitor. Out of this, the megakaryocyte or erythrocyte progenitor are mainly involved in the need or it gives rise to RBCs and the platelets. So RBC do not play much role in the immune defense mechanism because it is needed for oxygen transport. But nowadays, we started to understand the role of platelets in the immune defense mechanisms as well. Now, if we talk about the granulocyte, the macrophage progenitors, so this gives rise to all the granulocytes which we see in the blood. That is the neutrophils, eosinophils, basophils. Then we have the monocytes. And when it goes inside the tissues, it gives rise to the mast cells, and from the monocyte gives rise to a monocyte-derived macrophages in the tissues. So you see all these cells, they are involved in the innate immune mechanism. So all the cells of the innate immune mechanisms are derived from the common myeloid progenitor. Now, what the common lymphoid progenitor gives rise to? It gives rise to the lymphocytes, the B cell and T cell, and also the NK cell. Okay. So this is very interesting because we know that the NK cell, or for that matter, the innate lymphoid cells, which comes from the common lymphoid progenitor, these are the cells which are involved in basically the innate immune response as well. But the T cell and the B cell, they're the archetypal cells which are involved in adaptive immune response. So this goes and populate the lymph nodes. The lymph nodes, they mature further and give rise to effector cells. The B cells give rise to the plasma cells, which produces antibodies and releases it into circulation. And the T cells give rise to the activated T cells, like the T helper cells, T cytotoxic T cells, things like that. And NK cell can give rise to the activated natural killer cells. Now we have this another very interesting cell, that is the immature dendritic cells. Now, where this immature dendritic cell comes from, we know that the immature dendritic cells, they actually they take the information and from the innate system and passes it to the adaptive system. They're the postman which connects as the takes the letters from the innate house to the adaptive house. So these cells, now we know that they usually have a debatable origin. Some says that it comes from the mesenchymal cells, or some has it is from the common myeloid progenitor cells, but it is still a debatable thing. But this actually it is the region between the innate and adaptive immune system. So though it has been put into this white, a yellow bar over here, which goes to tissues, forms the immature dendritic cells, and which was a mature dendritic cells. So this is how the origin of all the vertebrate immune cells, which originate from your hematopoietic stem cells in the bone marrow. So when we talk about the immune system, it mainly comprises of two arms. One is the innate immune system, and another is the adaptive immune system. The innate immune system, we know it's present from the birth. Specificity is pre-programmed, and it includes non-immunological cells also, like the skin and the cilia. So when we say that innate immune system consists of the physicochemical barriers. The physicochemical barriers includes the epithelial barrier, and along with that, the mucin, which covers the epithelium. And at the same time, we also have a microbial barrier, that is the microbiota, which is also a part of the innate immune system. And along with these physicochemical barriers, we have the various cells of the innate immune system, so which we just talked about, at all the granulocytes. And at the same time, we have the sentinel cells in the tissues, namely the main three sentinel cells, which we talk about in the tissues are the mast cells, the dendritic cells, and the tissue-resident macrophages. And all these cells of the innate immune system are basically, they recognize the certain motifs of the proteins or the molecules, which is not there in in the human and in the human system. And what are these motifs of the molecules? There are certain conserved molecules which is present in a organism which is required for the organism's sustenance. Is required for
मैं तो कब से बैठी हूँ आपको जाना है तो जा सकती है
Please
So-
mute your mics.
Okay, so thank you. Okay, so this is basically this is the conserved motifs.
ऑफ करने जाएंगे फिर डाउन जस्ट
Dr. Shavani Shadan,
प्लान है कल होगा वन और
hello.
का कल फेटोस्टमी प्लान है तो ऑलरेडी सेंड किया हुआ है हाँ
It's a request to everyone to kindly mute your mic. It is causing a disturbance in the middle of the class. Thank you. It will be highly appreciated. Thank you, sir. So you may continue, and sorry for the disturbance.
Okay, thank you, thank you. Okay, and so basically this physical chemical barrier, it helps in invading of the 99% of the organisms which comes in contact with our body. And when there is a breach in the physical chemical barrier, this actually the organism goes inside. So apart from that, we have the various cellular and the humoral components of the innate immune system. There is adaptive immune system. Adaptive immune system, they develop during the life, and with exposure to the infection, the adaptive immune system kicks in. And this adaptive immune system is basically comprises of the lymphocytes. We talk about the cells, as the T and the B cells, and we talk about the antibodies, which are the humoral factors of the adaptive immune system. So if we see that the first line of defense is the innate immune system. And so they're selective, but they're non-antigen specific. And this innate immune system is a gatekeeper of the adaptive immune response. So initially, the rapid response is by all these cells of the innate immune system. It could be a mast cell, dendritic cell tissues, and macrophages, or it could be all the granulocytes, or maybe natural killer cells. And the humoral aspect we have talked about is like the complement proteins or the lectins and or the natural antibodies. And then if the infection persists and the innate immune system is not able to contain the infection, then they send the message to the adaptive immune system. So adaptive immune system comes into action after some time, usually after 224 to 48 hours, maybe 1 to 7 days. And they usually comprises of the B and the T cells, they're antigen specific, and they have a memory. So if we say that immune system is basically comprised of the innate and adaptive, innate always, we understand again, reiterating the same thing. The immediate response to a wide array of substances, it is non-specific, the skin and the mucosal membranes are there, and we have the cells and the chemicals, that is which forms the part of the innate immune system. Adaptive, again, the T and the B lymphocytes, and the plasma cells are the antibodies. Now, the basic mechanism of the innate immune system are two. When we talk about, one is the inflammation and another is the antiviral state. So the components of the innate or immune system we see, again, this we know that we have seen both innate and adaptive system has a cellular and a humoral components. Now let's discuss in brief about the innate immune system. So what are components? Number one is a physical and anatomical barrier, and along with that, we have also talked about the microbial barrier. Then we have the phagocytic cells. They are the macrophages and the neutrophils. And we have the humoral factors, which are the cytokines, the complements, the lectins, and the natural antibodies. But what are the eyes of the innate immune system? They are the cell surface receptors, and also we call it innate immune receptors or the pathogen sensors, which is needed for basically the phagocytosis. So the skin and epidermis, so they are mainly at the immunological forefront. If you can say that this is the chemical barrier, which is because of the pH and with the lipids and various the antimicrobial proteins which are secreted. And this is the layer which is there on the skin. This is skin landscape that is we're just talking about. This is the epidermis. Then we also have the microbial barrier, that is the skin microbiota. Then this physical barrier consists of this epidermis. And then below there is the dermis, which consists of these various cells. That is the dermal dendritic cells, the macrophages, the mast cells, and also the other cells also, they're important. So this is first the skin and skin epidermis. So when we see that the various cells, which is the innate immune response, one is a mast cell. We know that what does this mast cells do? These mast cells, they actually have the innate immune receptors, which are there on the surface of the mast cells, which are there in the cytoplasm of the mast cells. And these is receptors, one of the archetypal innate immune receptors we talk about are the toll-like receptors. Now, what these innate immune receptors, or for example, toll-like receptors do, they will recognize the certain structures in the microorganisms which is not there on the human cells. Now, what are these chemicals? These chemicals, for a bacteria, it could be the peptidoglycans, it could be the flagellins, or it could be the lipopolysaccharides. So these are the molecules which are recognized by these innate immune receptors. And if it is a virus, then it recognizes the nucleic acids. It could be a single-stranded RNA, double-stranded RNA, or a double-stranded DNA. So these are the molecules which are unique for that particular organism. It is recognized by the receptors, that is innate immune receptors, for example, toll-like receptors. And what does the mast cells do? When the mast cells is recognized as these as foreign organisms, so because of the receptors, it gets activated, and activation of mast cells causes the release of the mast cell granules and the granule contents. The granule contents has like immediately there's a histamine, there's a kinase, there's a tryptase, there's platelet activating factors which are released, and there is a rapid generation of the lipid mediators, as like the leukotrienes, prostaglandins, and there is also the synthesis of the various cytokines, like the various interleukins which are released, and this all aids in killing the organisms, and at the same time they contain the infection. Then we have the cells of the granulocyte, that are the eosinophils, that is the neutrophils. We talk about the neutrophils. The neutrophils, the main function of the neutrophil is the phagocytosis. So what they will do is that the neutrophils engulf the organisms by sensing it through various receptors. And after engulfing them, it kills the organism. And at the same time, after killing the organism, neutrophils also gets killed. And it leads to the formation of the pus. Okay. So this is what the neutrophil do. It comes into action in the first to come into the action, and then it causes the phagocytosis. Then there's the eosinophils. Eosinophils, we all know that the eosinophils have lots of granules inside, and eosinophil activation leads on to the various of these granules and the cytokines, and which is basically needed for cytotoxicity and also for the allergy. The macrophages, they are important not only for the killing the organism, they're the big eaters, but at the same time, they also eat up and clean and clean up.
Dr. Shiv Kumar, please mute your mic.
and at the same time, this macrophages also clean up the area where there's inflammation and maintains the tissue homeostasis. Natural killer cells mainly involved in the cytotoxicity because they have the receptors with which they can and actually recognize certain altered receptors on the various cells, and it can lead on to the release of the various as granules, also like the porphyrin and granzymes, and leading the killing of the cells. Innate lymphoid cells are a very interesting type of cell which originates from the common lymphoid progenitor, but they do not have the gene rearrangement and the receptor, like the B cell receptors or T cell receptors, they do not have, but they have the receptors for the innate immune receptors, and they are involved in the early phase of the initiation of an inflammatory cascade. Dendritic cells, we all know, they are not only involved in activation and release of the cytokines and containing the infection at the tissue level, but they also carry the organisms or the peptide information from cytoinflammation to the draining regional lymph nodes and activates it's the naive T lymphocytes over there. So how does the entire thing starts? So innate immune system recognizes, I have told, pathogen associated molecular patterns or the pathogen related molecular patterns. And these molecular structures are the microbial pathogen that require for survival, namely the lipopolysaccharide, peptidoglycans, flagellins, teichoic acids, and things like that. And for the viruses, it's the viral nucleic acids. Or it can recognize certain damage associated molecular patterns, and these DAMs usually causes a result of the cell damage by the infections, and this releases certain molecules which are actually recognized by these receptors. And this pattern recognition receptors are basically that we have talked about. There are many. And now, what do they do? Now, these pattern recognition receptors, they actually, there's several families of the proteins. They express on all the effector cells, and most importantly, provisional antigen-presenting cells like the macrophage dendritic cells and the B cells. They could be a transmembrane PRRs, cytosolic PRRs. Now, we can say that the receptors.
Dr. Sayan, please mute your mic. Dr. Sayan, please mute your mic.
Industry of
Okay, so the principal functions by receptors, one is the opsonization, that is, they basically helps in actually coating the organism, so that they becomes more palatable for the phagocytosis. They activates the complement, they cause the phagocytosis, and activates the pro-inflammatory signaling pathways, induce apoptosis. Now, there are mainly three classes of the pattern receptors. Most common that we talk about is the signaling PRRs, of which we told like receptors are the archetypal signaling PRRs. We also have endocytic PRRs, which are their endocytic vesicles, and these are mainly involved in the recognition of the viral nucleic acids or the viral proteins, or we have secreted PRRs, which are released inside into the circulations. So very important to understand that there is this unique actually this circuit of the monocyte macrophage and the dendritic cell interactions. And this interaction of the dendritic cells or the monocyte and macrophages is basically meant to actually kill the organisms. How this interaction takes place? This interaction takes place by the release of the various cytokines from the dendritic cells. So that means the dendritic cells, when they get activated, they release cytokines like the TNF alpha, the interleukin 1, or the interleukin 2, and which is going to definitely activate the macrophage and cause it and helps it in killing. At the same time, the macrophage, it releases the same cytokines into the milieu, which causes the activation of the dendritic cells. So this bidirectional activation of both the cells, it's important in amplifying the mechanisms of killing the organisms at the tissue interface. And we talked about the natural killer cells, and so how it is involved in the killing of the cells which is showing the altered receptors on its surface. Mainly it is important for the immune surveillance and prevention of the development of the cancers. So, very, very important and component or the effector part of the innate immune system is the phagocytosis, which is important and for actually containing the organisms at the tissue interface itself. So we know that the professional phagocytes are the macrophages, but apart from that, we have these macrophages which are derived not only from the monocytes, but we have tissue-resident macrophages as well, which are also derived from the monocytes, but they go and home in the particular tissues, yes, and they stay over there. So this if and this is required by various cytokines or various factors which cause the homing of the cells, the particular tissues, and so that they can attain a particular morphology. So just like the tissue-resident macrophages in the brain, it is known as the microglial cells, which are professional macrophages in the brain. If it goes in the lungs, we call it the alveolar macrophage. So if it goes in the spleen, we call it splenic red pulp macrophage or marginal zonal macrophage. The peritoneal cavity, they are called peritoneal cavity macrophages, and in the skin, they are called the Langerhans cells. And you see, these are the various cytokines which is needed for this is tissue macrophages to home over there, and these are various transcription factors which are required and for actually making this particular phenotype of that macrophage in a particular organ. So this is the phagocytosis. This is central to the innate immune function. This is required for the inflammation. So we see that this is a microbe, and this is basically phagocytic cells. And these are receptors, which could be a mannose receptor, or it could be integrin, or it could be scavenger receptors, and with which the microbe is opsonized, and it is then internalized and then forms a phagosome. This phagosome then actually it combines with a lysosome, forming a phagolysosome. So all the digestive enzymes are released into the phagolysosome, and it's partially digested. The pathogen residual bodies there, and the residual materials are then by exocytosis, they are eliminated from that. So this is the usually the process of the phagocytosis, which is very important for the innate immune function. This under the microscope, you can see how this as phagocytosis is taken place. It is the organism outside taken inside, and then it is entirely destroyed. So the effector mechanisms which are attributed to the phagocytes, it is important for the killing of the microbes. This is important for the inflammation enhanced activity of the immune system, and also the tissue remodeling. And these all the signaling in molecules which are released by the macrophage are important for that. So just a little bit about the pattern recognition receptors, because nowadays this is playing important roles in the various disease states. So always we understand that it could be cellular PRR, like which is actually there in the cell surface or in the cytosol. Also, or it could be humoral PRRs. Humoral PRRs are mostly we talk about the collectins and the pentraxins. And one of the most common pentraxin that we all talk about in a day-to-day clinical parlance is the C-reactive protein, which is a pentraxin. And the cell surface receptors are the phagocytic receptors, as like it could be scavenger receptors, it could be C-type lectin receptors, or it could be sensing receptors, like the toll-like receptors, which are transmembranes. So these are the toll-like receptors, and these are C-type lectin receptors. So they're all helpful in causing sending the signals inside the cells or the innate immune cells, and leads on to specific activity of that particular cell. So these are various PRRs, as we had already talked about that. Like it could be not only TLRs, or it could be a C-type lectin receptors, it could be a NOD-like receptors, it could be a RIG1-like receptors, and RLRs, all those things. So though there are 13 subtypes of the TLRs, we know that TLR 3, 7, 8, and 9 located in the intracellular vesicles, and they're usually important for sensing the nucleic acids of the viruses. 2, 4, 5, 6, and 10, and 1 located on the cell membranes are mainly important for recognizing the bacterial PAMPs. What the TLRs do, TLR activate through this very common pathway, that is NF-kappa B pathway, and it causes release of the pro-inflammatory cytokines like the interleukin 1, beta, TNF alpha, and interleukin 6, which causes the activation of the surrounding cells, including the macrophage, as we have just discussed before. So this activation of the TLR leads to release of various cytokines, which can subsequently result in the activation of the adaptive immune system. And very, very important, one thing I would just like to tell you that it is the type of TLR which is activated, it will send the certain kinds of the cytokines and make the change the milieu. And in the changed milieu, it accordingly will give the information to the adaptive immune system and to move into a particular direction. And how which TLR will get activated depends upon the which microorganisms. So it is the particular microorganism which, by activating a specific TLR, this activates a specific downstream signaling pathway and causes release of certain specific cytokines. And the specific cytokines as actually gives the direction to the cells of the adaptive immune system to develop into a particular type of the adaptive immune response. It could be a Th1 response, it could be a Th2 response, it could be a Th9 response, Th1722 response, or it could be a TREG response. Okay. So, and macrophages, there are plenty of the receptors. They're all innate immune receptors. And mostly this is just overview to make you understand that how this cell sense. These are the eyes. They have numerous eyes, and with which they can actually see so many molecules, and they can sense it, and they can just react accordingly. So what happens is that it is microbe, it goes. So whatever the immediate would be the resident cells, which are the first we have the antimicrobial proteins and so like the dectins and all, which tries to neutralize the microbes, or we get the help of the phagocytes, macrophages, epithelial cells, innate lymphocytes, and mast cells, which try to contain them. And these also releases the cytokines. The cytokines causes the endothelial cells to become more porous, and the cytokines also helps actually in causing the cell adhesion molecules to be expressed by the endothelial cells, and this causes the various cells of the granulocytes to actually come into that particular site. So these are the chemoattractant molecules and the cytokines, which causes the other cells of the innate immune system from the intravascular compartment to come into the tissues and fight the infections. This could be neutrophils, monocytes, eosinophils, basophils, or it may be the other cells. And these cells are also the same time they're killing the organisms and releasing the interferons or the interleukins. And these interleukins further it causes the activation of the adaptive immune system. And at the same time, there is the dendritic cells also presenting in the peptide to the adaptive immune system. So it causes the adaptive immune system to mature. And once the adaptive system is matured, this adaptive immune system then actually releases the further cytokines. If it is a Th1, and it will release interferon gamma. This interferon gamma is going to instruct the macrophage and increase its effectivity for killing. So there's an instruction from the adaptive to the innate comes after it is activated. And entire when this fighting is going on, but at the same time, at a certain period of time when the infection is controlled, the homeostatic mechanisms comes into play. And all these, as regulatory lymphocytes, epithelial cells, dendritic cells, monocytes, macrophage, all are important in bringing the homeostasis in the tissues, and they impact the innate immune system, the cells of the adaptive immune system, and also the instructing cytokines going to the adaptive to innate and maintains the tissue homeostasis. So this is a very interesting schematic diagram which shows and all. So NK cells, we have talked that it is important. Natural killer cells are important for the immunological surveillance. That is, they have the receptors, the cell surface with which they can recognize various cell surface receptors. And if they recognize that, they cause the release of various molecules, which are called the porphyrins, which can form a transmembrane pore or the granzyme, which initiate the apoptosis of the particular cell. We come to another important and means function of the innate immune system is the antiviral state or the antiviral response. So what is the antiviral state? Now, what happens is that when and there is a viral infection of any cell, so it could be epithelial cell, or it could be a specific type of dendritic cell, which we call the plasmacytoid dendritic cells. Now, when these plasmacytoid dendritic cells are activated, they causes the release of a specific type of interferons, which are called type 1 interferons. Now, all the cells have the receptors for the type 1 interferons. So when this type 1 interferons, it binds and to the type 1 interferon receptors of an uninfected cell, it causes a certain downstream signaling and causes the formation of certain molecules, which are called the RNases, or the activation of the RNases. Okay. So what does RNases do? Now, see, RNases, if the cell is not infected with the virus, it is not going to do anything. So it's just going to be like that. But if the cell is infected with the virus, then the virus, it undergoes, it goes and combines with the host DNA, and initially it forms a single-stranded RNA. Now, the single-stranded RNA of the virus is an important obligate, it is an intermediary. It is a nucleic acid in the viral biogenetic pathway. And this obligate single-stranded RNAs, if they're present, the viral RNAs, if they're present in the cytoplasm, they will be cleaved by these RNases, which is this activated RNases, which is caused by this as pathway of the interferon receptors. So that means it in this way causes degradation of viral RNAs, but at the same time, it also causes inhibition of the viral gene expression, virion assembly, and the viral proteins synthesis. And in this way, what happens is that there is this type of interferons, which forms a shield around the non-infected cells and prevents it from the viral infections. And those cells, which is already infected, it causes the degradation of the RNA inside and prevents the infection to spreading out. This is called the antiviral state. Now, what are the antimicrobial proteins which are secreted on the cell surface? Like these antimicrobial proteins are secreted on the skin, they are secreted on mucous membrane, they are secreted in all respiratory tract, GI tract. They are basically, they has multiple functions. They can directly inactivate the organisms. What we talk about commonly is defensins and the catalycidins. And they will usually just go inside the cell membrane and cause the formation of holes and the lysis of the cells. So these are the other types of humoral part of the innate immune system. These are the collectins. The collectins are the macromolecular structure. They usually bind to the microorganisms, and by binding to the microorganisms, they also activate the complement pathway and causes the lysis of the organism through the lectin pathway. But most important collectin we talk about is the mannose binding lectins. The very important component of the humoral aspirant immune system are the complement system. We all know that complement system has multiple functions. The most important protein in complement is all secreted by liver is the C3. And the activation of the C3 can occur by spontaneously, that is when it comes C3 in contact with the bacterial cell surface, or it can be by the lectin activation pathway as we have just seen, or when it also forms a part of the adaptive immune response, it is through the classical pathway activators by the antibodies. By whichever pathway, the C3 is cleaved into a smaller particle C3a and a bigger particle which is C3b. So the C3a is important in causing, goes and binds to C3a receptors on the microbes on the cells, and makes it more palatable to be phagocytosed. And so this is also anaphylatoxins. So C3b, it has a C5 converters activity, and it breaks the C5, the next protein, into C5 and C5b. So the C5a also acts as anaphylatoxins, just like C3a. And the C5b, it binds to C6, 7, 8, and 9, forming a big complex, which is called a membrane attack complex, which can go and just drill holes inside the cell membranes of the microorganisms and just kill them. So this was in very nutshell and very brief about the innate immune system. So shall we discuss adaptive immune system in this class, or we'll take another class for the adaptive immune system? And we have 10 minutes, we can have some Q&A on innate immune system, or means what do you say? Hello?
Yes, sir.
So shall we have the adaptive immune system another class? And today
Yes,
Yes, sir,
sir.
sure.
Yes, sir.
the innate immune
Yes,
system, and we just take some questions if somebody has any.
If any aspirants have any question, you may ask now. So I think for the time being, they do not have any question. If there is, we'll discuss in the next class.
Okay, thank you.
Thank you, sir.